video unless you want to see a face during your talk. I don't care. Yeah, so I'll hand over.
Right. Thank you. Thank you for the invitation and thank you for the kind introduction. Okay, so today I will talk about EEG and EEG reproducibility. So let me share my screen. Okay, so I will illustrate using real EEG data how we can basically write a script and relatively easily so that we can have a fully reproducible workflow. And I'll do it here using EEGLAB and also some tools that we developed, LIMO EEG. Just to give you an idea of the workflow and also the way the talk is structured, which reflect that workflow. So we start with EEG BIDS. So BIDS is brain imaging data structure that allows you to organize your data. And then because it's organized in a way which is very structured, we developed what so-called BIDS tools, which allows in one go to import all of the data, all the information that's needed to process the data. And then from that, we'll move into EEGLAB, and I will present mostly some recent development about ICA and machine learning, and then how it's implemented and how EEGLAB and LIMO EEG work together to, in 3 lines of code, do a full stack hierarchical linear model. Just to make myself clear, so this workflow represents a sequence of computational steps. And that's basically what we mean by workflow. And that's different from a pipeline, which is what actually I'm going to show for the rest of the talk, which is the set of data processing elements which are connected in series, and each element can be processed in parallel or not. And that's the pipeline is what implements workflows. Okay, so let's start by the EEG BIDS, the first part. And I'm going to show that using the data that everybody can download, which are the multi-subjects, multimodal human imaging dataset, which was a simple experiment. So we've got MRI, EEG, MEG, all sorts of things. We're just going to focus on EEG today, but the experiment is simply to present 3 types of stimuli: famous faces, non-famous faces, and scrambled. They're repeated at different times, so first time, right after, or much later, which basically allow us to look at priming effects. So we start by the data, and for that we look at the brain imaging data structure. So I'm going to spend a little bit of time explaining BIDS for those who are not familiar with BIDS, because it's an important piece of work that's been done for the last 5-6 years now. So BIDS is a data structure, which means that's nothing to do with format. Someone has their microphone open, if you could turn it off. Thank you. So it has nothing to do with the format. The format is a typical format that we use. I'll come back to that in a second. So BIDS is mostly about how you're going to organize data in a folder, how you name your files, how you document your metadata. And all of this is done using community standards, and that's really important, as well as dictionaries. And so here's an example that you will see. It's on my hard drive right now. For this particular experiment, I've got my subject, which are called sub, and I've got something which is data descriptions. If I go inside the subject, for instance, subject 2, I would have my anatomy, which is a typical Nifty file, and I've got EEG data as well. Now you can see there is name redundancy, which is typical things in BIDS, which makes it harder to make mistakes because now the folder is called sub2, inside sub2 are not, and it's also still called sub2. And then you can see it's always JSON files, which are the metadata describing the data. Each dataset comes with a JSON file data description dataset, which basically makes your data easily identifiable and citable. Now, the participant TSV is a standard things, tabulated separated values. So that's your subject information at the root, easy to figure quickly the age, the populations, demographics, and so on. Now, if we look specifically at EEG BIDS for today, so the EEG BIDS is an extension of BIDS. So we developed the standard specifically for EEG. And so if we look now into that subject 2 EEG, for instance, we can see I've got subject task recognition, we've got electrodes, we've got the different runs, even TSV. So typically for MEG, all the formats supported. For intracranial EEG, we've got EDF, GDF, and FIF. And for EEG, we use mostly EDF and also VHDR, which are the two official versions, but we also take some sets and BDNF. Specificity of metadata. So for instance, if we look into what I have a task channel TSV, which will describe all the EEG channels with the unit and the type of channel that corresponds to this. There's also the electrode. So the electrode, remember, is the physical things that we put on people's head, whereas the channel is what is in the amplifier. It's not the same thing. That's why it's separated files. The electrodes come with positions. We also have some information about the task itself, about the positions. And then, of course, we've got the event TSV. So the event TSV describe over time all the different trials, the different codes, and so on. So this is particularly important because, of course, this seems like something which is redundant when you prepare your data. But typically, we use triggers, so that comes up as a number, it doesn't come as a name. And it's also much easier to figure out for users because it's a simple text file. So once the data are prepared using BIDS, which is pretty much every data set now that we share are done using BIDS, we can use for EEGLAB BIDS import, so the BIDS MATLAB tool. And you can see here, of course, it comes with a GUI, which is typical thing in EEGLAB, but it's also a single line of code. So we put pop import BIDS, we put the file path, like where is the root of that BIDS directory with all of the data, possibly an output directory, and in one go, so that's the data as they are, and in one go, that would create in EEGLAB a study and make a copy of those data. In EEGLAB, a study is typically a structure that contains all the information related to data preprocessing and analysis. That means that if you have data which are in BIDS, with one line of code, you can import all of the data, and that will contain as well all the information about the triggers, the channel locations, the anatomy, if you've got the T1, and so on and so on. Which makes things very easy because now I don't need to try to understand the structure of the data. I simply have to import it, and I already know and I already have all the information I need. Now, if your data are not structured in BIDS and use EEGLAB, you can just as easily create a study. Well, it's not as easy, but you can create a study and in one click export it into BIDS. Okay, so that's the first part of the workflow, which really is very easy because it's a single line of code and you can import data assuming they are prepared in BIDS. Now, the second part is the pre-processing of the data. And so we have typical recommendations about that. published this year. And this is done in EEGLAB very easily. Basically, you see the top of the, so that's the script for the whole data pre-processing of the 18 subjects. And that's it. There's nothing else. So we can see at the top, we've got my import BIDS. Then the two main things that we have now in EEGLAB, which makes things very easily, is this pop clean raw data and also AC label. So what, of course, you can use whatever you type of functions you want to pre-process your data. What I want to focus on here is the fact that we want automated data pre-processing. So that doesn't mean that we should not look at the data and apply blindly algorithms. You probably want to tune your parameters and choose carefully. But it's important to have this kind of fully automated data pre-processing, and it's necessary for reproducibility, because this alleviates variability that you have if it's user dependent. And so that's typically what was happening when using ICA. So ICA is a great tool to do data decompositions, and especially for artifact rejection. But you know, before having IC label, that was up to the users to define which components were brain data and which components were artifacts. So let's look a little bit into this IC label. So automated AC labeling, typically 2 lines of code. One, you run your ICA, right, whichever ICA decomposition you fancy, whatever method you like. Then we call the IC label, which basically is the algorithm which does automatically the classification, and for each component coming out of your ICA, it will label it, and then pop subcomponent will simply reject the components that you want. So the way it was done was to use a classifier, a multidimensional classifier based on the topography, the time course, spectrum, and so on. And the classifier trained on thousands of crowdsourced labeled ICs and hundreds of thousands of unlabeled ICs from the Schwartz. And so the way we ask people to label, so there was expert labeling, but also people not necessarily experts. And so we present different ICs, and each time people have to choose whether it's brain, muscle, eyes, or flying, channel noise, or even something else. Once we've trained the classifier, we can throw in new data. So here's the data from one subject, for instance, using this phase dataset. And these are 2 components. First component IC6 and another IC10. Both of them with some activity coming from the right inferior temporal cortex. What's interesting is that IC label, for instance, here you can see, because we have a nice dipolar configuration, is labeled nicely, 99% chances to be brain data, whereas this one is labeled as being mostly muscle. And so because now we have a probability assigned to each component, it's very easy to say, for instance, let's accept every component above 90%. And so this is what we've done. And now we have data which are imported, automatically cleaned, and we are ready for statistical analysis. For that, we use the LIMO toolbox, which is now fully integrated with the EEGLAB and the EEGLAB study. And so very quickly, if you're not familiar with this framework, it's a hierarchical linear model, which means for each subject, we use a general linear model where we express each time point at each channel as a function of the design matrix, which is your experimental conditions, for which we obtain beta parameter estimates. And then we can throw in these beta parameter estimates in the second level, for which we use different statistical tests, and then apply corrections for multiple comparison. Now, this has been completely integrated with EEGLAB study. And now all the events were defined at import, right? Because the data were prepared using BIDS, which means using BIDS, we already knew which part of the signal contained the different experiments, and automatically it allows in study also to create the different experimental conditions. And so that's just to show you, for instance, how it would look like for the GUI. And in 3 command lines, we can do a full hierarchical linear model. Those command lines are that simple. First, from the study, we create a design. So for instance, here I use the 9 experimental conditions, which are, if you remember, we've got famous faces presented 1, 2, and 3rd time. Then we've got scrambled faces, and then we've got unfamiliar faces. And I'll do that for all my subjects. So that's the first line of code we want to do, create the design. Then we simply call pop limo, and that will basically compute the linear model for every subject. Once we've got the parameter estimates for those 9 conditions, we can call limo random select. Here I want to do a repeated measure ANOVA, and I want to do a 2 by 2 faces versus repetition. And that will compute the ANOVA across all the subjects. And if we do so, we obtain this type of results. So we've got all the channels on the y-axis, the time and the x-axis. The data are corrected for multiple comparison. What we obtain is a significant effect of a type of faces, which replicates, as we can see here, the results published by Wakeman and Hanson, the typical difference where real faces elicit a stronger N170 than scrambled faces, and also this later difference. And we also have a significant effect of repetition that we can see over here without interaction. Now you might think it's also a bit strange to have no interaction, but in this type of design, that's not what you can see. But just as easily as we can do an ANOVA, because it's a linear model, still with this same 3 line of code, we can create a little bit more complex model. So for instance, here I use famous, scrambled, and unfamiliar faces. But then I use a continuous value, which again is defined using BIDS import, which corresponds to the time between each level of presentation. So how long between the first time you see a face and then the 3rd time. And then again, we compute the model, and then we estimate a repeated measure ANOVA, but this time simply based on the parameters, which is the time difference. So again, this is the design matrix for each subject, the 3 types of faces, and then the distance between the 1st and the 3rd repetition. Well, the 1st and 2nd, and 1st and 3rd. And I'm interested in only those 3 parameters there, which is the time. We can see a significant effect later, around 550 milliseconds, which basically we have a significantly stronger modulation of the ERP as the time passed. And it was only the case for famous faces and not for unfamiliar or scrambled faces, which means we do have an interaction. But of course, it's much more complicated than a categorical effect because it depends on the time that elapsed. And so this illustrates that it's worthwhile spending time to have all sorts of measures for your stimuli and to put that in your event TSV file, because it's very easy then for everybody to, or for yourself to create multiple types of design. Here it's a one-step integration of the effect. And it's useful because it allows you not only, for instance, in this case, to do something which allows to test an effect, but it also could be very much like confounds in your data and your stimuli that you know exist and very hard to control as you want to control for. So what's the conclusion of all of this? Is that yes, we can. What can we do? Well, it doesn't have to be a very convoluted script. As you could see, it didn't take a lot of lines of code. It does not require human interaction. Once it's scripted, we can fully reproduce all of the analysis and the figure. So from importing with BIDS import to the figures that are presented here, it's about 200 lines of code as presented in that GitHub repository. And it's also now a paper in press. And also simply to highlight that, of course, it's work that I've done with people at the charts from EEGLAB, and also part of a series where not only for EEGLAB, but it's also possible to do it with many other tools which are part of this research topic. Thank you.
All right, thank you very much, Cyril. Okay, someone's clapping loudly. I don't know who it is. Jeff.
Well, that was me, yeah.
Okay, so yeah, we've got a lot of participants here, so I bet there's going to be quite a number of questions. Feel free to turn on your video, unmute, and ask Cyril anything you might be interested in. Yes. Go ahead. Micah, do you want to just go ahead and ask?
Sure. First, thank you very much, Cyril. Always nice to hear you talk. I was wondering if you could talk a bit more about the features in particular that are used in the statistical modeling and what limitations there are or aren't in your pipeline.
Well, I mean, for this data, that seems to be the right type of model. It's linear time invariant, right? So it means you apply the same model every time point. So it could be so channel time frequency or time frequency if you do time frequency, which means it's not accounting for overlapping responses because it's not a model over time. Otherwise, limitations, you know, that's pretty much it. It's the limitations of linear models.
No, I meant but, more. You're looking principally in sensor space.
Now we can do it in ICA space, and right now I've got a student who's finishing the work for source space.
Okay.
It doesn't really. I mean, the models by themselves, you can apply them in any space you want. It's more an issue of how you want to correct for multiple comparison. Mm-hmm. So for IC space, at the moment, the only comparison we have is based on maximum statistics. We do have some other ideas, but it hasn't been very successful at the moment. So maximum statistics always work, right? The idea is simply you generate data under the null. Since your family-wise error is the probability to make at least one error, if you take the biggest effect under the null and you control for that biggest effect, then therefore you control for the entire data space. Now you can apply the same thing, but using, for instance, clustering, which works well for source space and for sensor space. But for IC space, you know, because they are supposedly maximally independent, but they are not independent. They're only maximally independent. Yeah. And so you might want to say, you know, there's some relationship, but it's difficult.
Yeah, I was thinking more in terms of some of these global measures, which, in my opinion, would allow for more reproducibility across labs. Because you get away from some of these reference-dependent measures, you get away from these sort of single-channel analyses that I think cause a lot of problems in the literature.
Here, it's not single channels. Here, it's just the whole data space, right?
Yes and no.
Well, what do you mean? Yes and no. I analyze the whole sensor space there.
Yes, and that map of statistical significance applies at individual sensor level. You correct for multiple comparisons, sure, but the interpretability at the end of the day is at the level of the single sensor.
That depends on the. It's not what I do. Because if my statistic is a cluster statistic, then I don't interpret at the level of the sensor, because that is not what my statistic is based on.
That's right, but it can lead to confusion,
Yeah, but,
as you can see. And
people do not understand statistics.
That statistical
it's a
result,
issue.
the statistics you're showing are valid for the choice of reference that you put in. So if you change the reference, you'll get a different result.
Yes, I've got another paper on that.
That's why I'm curious if the objective is reproducibility, should we not move to reference-free measures? That could be the Laplacian, that could be the topography, that could be the global field power. I'm not trying to push a particular agenda.
You're talking, Mika, of replicability. I'm talking reproducibility. I can redo the same thing, and if you look at the effect size, and we've done that with 12 different pipelines. The effect sizes are very similar, by even changing the reference. Now, if you're talking about replicability, when you threshold, yes, this is much more variable. Because the variance is very much different.
Exactly.
Not so much the raw effect size, but the variance, especially some referencing frameworks just give very weird things. I agree with that.
Now, that said, I mean, I think the standardization that you're doing is fantastic. So I was no means trying to suggest
No, I understand that. I was just trying to, I think it's because also there's a difference between reproducibility and replicability. And I agree that if we look at for instance, sensor space and different referencing scheme, the replicability is much more different than the reproducibility.
Sure. But that's reproducibility of doing the same analysis on the same data set. That's a different kettle of fish. But I should stop because there are other people that want to talk besides me.
I think I'm the only other hand up, so I'll go. Thanks for the talk. It was really interesting. And frankly, I quite appreciate this stuff because it does save a ton of time in coding and sort of writing a whole pipeline when you have these things kind of preset and you really can't just jump in and get this stuff done in a very small number of lines of code. And actually, just to kind of sort of circle back to what Micah was just mentioning, because I'd had this conversation with you, Micah, maybe a month and a half or two ago, about some RSA techniques that I was developing. So one of the easiest ways to make sure your data are reference independent is to baseline them all with respect to some other condition. That way, no matter what reference you're subtracting out, the difference between any two conditions that have the same reference is necessarily reference independent, in the sense that like A minus some reference compared to B minus that same reference, that reference is now sort of subtracted equally from both, and it doesn't matter what that thing was that I subtracted from both, it's the same. So no matter what reference you choose, once you're taking condition by condition differences with respect to a same reference, whatever it is, there you're sure that whichever reference you picked, the final result will look the same. But that's not always a clear-cut thing to do, in the sense that different designs might have different baselining requirements, or you might want different baselines for different conditions, in which case Micah is correct. And I'm wondering whether you've considered any plans for implementing maybe like baselining protocols.
Yeah, it would be maybe 2021, maybe. So basically for each channel, if you do in channel or for each source, for IC, I still don't know what to do, but for sensor space or source space anyway, each time we apply the model, we can, so instead of, you can have an extra, so now the model will be different for each channel or for each source, because it will be your design matrix plus the baselines that you want to subtract. Mm-hmm. So now the effect over time or frequency and source So there's already a paper like this, on this where we show that if you do the regression that way, so basically it's all regressions, right? General linear model. Then sometimes it's similar to do your normal, usual kind of baseline correction, but in some other channels, or it just doesn't do it because it's nothing to do with subtract, and your subtractions that you've done before, by forcing it in a way, you create differences which are smaller or bigger. So there's only one paper on this, but it's an interesting aspect because what you're saying is, okay, well, do the variations that I observe across all my trials now, because it's a single column, right? You don't do it specific to condition. So you would have, say, your 9 conditions plus 1 column, which is your baseline measure. You say, well, find me over time, where does that, do I have variations which cover that baseline? And if so, apply it, correct for it. Well, I mean, you still compute your parameter in fit, but this parameter gets smaller or bigger over time. And sometimes on some channel, it does it everywhere. Sometimes it doesn't. So it's interesting to see these corrections. It might be also easier to apply this type of corrections for time frequency, because it's quite hard to find a good baseline for time frequency. So this approach maybe for time frequency might work a little bit better. I don't know yet. It's something we just looked at a little, but too many things to do.
Yeah, I can imagine.
But yes, it's somewhere in the long to-do list, but towards the top, because it's something which is very interesting in terms of correction.
Yeah. Thanks.
You're welcome.
Here's another question from Mikael.
Yes. So thanks a lot for the very nice talk. I'm not an expert in EEG. I work mostly with MRI, but we are really looking into using BIDS and automation also with EEG. So I had 2 high-level questions. One of them is, could you comment on the stability and maturity of the EEG and MEG BIDS, current BIDS status, and how fast it is evolving, if we can already use it and it's relatively stable, or if we should expect quite a few changes? And the second question is regarding EEG. It seems, maybe I have the wrong impression, but there are often still manual steps in the pipeline. And what would be the best practice on how to formalize these manual steps so that to make the study more reproducible or replicable?
Regarding BIDS for raw data, it's fully stable. There's no changes for the last year. Now we're working on BIDS derivatives. So the raw data are fully stable. You've got tools in Python and in MATLAB. So there's no, no reason not to use BIDS EEG. You've got BIDS for MRI, PET, intracranial EEG, MEG, EEG.
So it's mostly the derivative section which is evolving quite rapidly.
Yes. I need to post the roadmaps that we made a couple of months ago. And, well, it's evolving rapidly. It's evolving. But that's derivative. So that's nothing to do with the way you want to do your data processing and raw data, because derivatives is the output of your data processing.
Sure.
But derivatives are useful for other aspects. They're also useful for aggregating data. They're useful for machine learning. They're useful for other aspects that we are interested in, but not for the reproducibility itself. Now, features which needs to be automated, well, I don't know what, like in the pipeline I showed you, there's 0 manual work.
Sure.
Well, I mean, the manual work is obviously to code, but once the code is done, that's it.
So in different set of electrodes, when you compare manual identification of artifacts with the automated method, no matter the number of electrodes, you have comparable performance? Or does it vary according to, I mean, headsets and?
So if we compare, for instance, AC label with Mara, which is another machine learning classifier, the results are fairly similar. Didn't see. I mean, the decomposition is always the same, right? So we use I use the same ICA, but just use two different classifiers. But we end up with a very similar set of labels. Now, compared to manual, I don't do manual because if it's me, you know, that's not going to be very good because I'm not very good at labeling ICs. If it's Guillaume, maybe much better. If it's Scott, Scott has been looking at ICs for you know 30 years. I'm sure his labeling is very good. I don't know. I mean, it really depends on who does the labeling. So I don't see really. For me, there's no point really comparing the automated versus. I'd rather compare my classifier performance compared to experts, which I know are experts, than me doing it.
Yes.
I don't know. I mean, you can also do everything without ICA. You know, it's over tools. But I think there's plenty of tools nowadays where, I think the point what I was making was to say that you probably want still to. It's not because we automated the tools that you don't want to look at the data in between, right?
Yep.
You probably want to do some optimization. So, for instance, I quite like recently I've been using a lot of Zapline as a way to remove the power line. But again, it does need some tuning from one data set to another, for instance, in the parameter sets. And so that's an issue because in a way you might want to justify it, but it's difficult to justify. But once it's there and it's in the script, you know that for sure people can redo the same analysis. And now the problem is, of course, if I do the same experiment as Mika was talking about, applicability, if we redo the same experiment with the same equipment, that's another source of variability in EEG. Realistically, the same parameters should apply, but we'll see with EEG reproducibility with the Minilab project, right? I don't know if you guys have seen that, but it's a big EEG Minilab project, and so we'll see with that what what difference we have.
Sounds great. And just you mentioned your article about this project. Is all the data and code open?
Everything will be open, yeah.
Okay, great.
The mini EEGLAB, the mini EEG project at the moment, we only published the plans and stuff like this. Data haven't been collected yet. But everything is on OSF and GitHub.
Great. Thanks a lot.
Okay, are there any more questions from those who are still with us? Yes.
Hi, I have a question. Quick one. So we're interested, we have like a set of data sets, all data sets that we would maybe be interested in formatting them as in BIDS. So first of all, is that possible? To have, like to do that with already collected data, and how flexible it is with like some missing information. Let's say we don't have from all data sets, maybe potentially all the information about the subjects or stuff like that. So
yeah, I mean, there's no issues there. I mean,
okay.
It's very usual to not have all of the information.
Okay.
So no, there's absolutely no. So I can't say much about like all of the tools, but the ones that we developed within the as a plugin for EEGLAB, if you just import the data as a study already in EEGLAB, then basically it's one click to export to BIDS.
Okay, that's great.
And if you have problems, because maybe in all the tools that we wrote, there will be some issues with missing data or something like this, but you can always send an email on the email list or an issue on GitHub and we'll fix it.
Okay, great, thanks.
Okay, so if there are no other questions, then I'd like to thank you once again, Cyril, very much. As I said, I will send around the video so that you can place it wherever you want on your website, and we'll link it, and I'll send the link to all the attendees here in this list. Thank you so much again, and thanks everyone for sticking around. Great talk.
Cheers. Bye-bye.
Yeah, thanks again.
Okay, have a good evening, everyone.
You too. Bye.
Merci bien. Au revoir.